So without further ado, it is my great pleasure to introduce our keynote speaker, Andrew Hopkins, who is the founder and CEO of Exscientia, one of the UK's largest and fastest growing biotechs. Andrew published two of the first papers on large-scale machine learning on pharmacology data in Nature Biotechnology in 2006 and 2007. In 2012, he founded Exscientia, the first company to pioneer the use of artificial intelligence to design new medicines based on the algorithms that he published in Nature. He led the teams that discovered the first 3 AI-designed drugs to enter human clinical trials. The title of his talk this morning is Redefining Medicine Through Patient-First AI.
Thank you so much. Excellent, thank you. It's a real pleasure to be here today, and an honor. Thank you so much to the organized committee for the invitation. And even though this is the first inaugural meeting, this is a field actually that's been in development for nearly 20 years. I was talking to Anne Carpenter just before. I remember Anne's probably first talk. She gave it a keynote, probably back in early 2003, something like that, when she was still a student, on the possibility of where cell profile was coming, of the use of computer vision in the analyzing of biological materials in high quantum microscopy. That concept then has stimulated multiple new companies, from in situ to recursion to all-site, et cetera. An entire new field now we'll hear about has developed from that. We have Pat here today as well, Pat from Relay. Pat has been one of the leading lights in computational chemistry for many years for his work. And now we're starting to see, just recently, the results of one of his first designed drugs from a technology using high performance molecular dynamics. Had a fantastic phase readout in patients, a phase 1b, by something like an 85% response rate, a highly targeted medicine. What we see now are actually sort of the foundations of where this field could go. And one thing I want to say is I think all of you sitting in this room are probably here for a single purpose in a way. And that purpose, I think, is the same one that actually led me to found Exscientia, which is a frustration with the way how medicines are created. Why does it take so much money? Why does it take so long? Why is medical need so huge? And yet, our ability to sort of drug the genome is still incredibly sort of limited and slow. In the nearly over 100 years since Paul Ehrlich first started and invented the field of pharmacology, we probably only drugged just over 1 or 2% of the human sort of proteome in a way. Only less than sort of 2,000 unique new medicines. So how do we accelerate that? How do we move from a position of there being drugs being a sparse resource to an abundance? And I think a key to that is actually using machine learning and AI. Why do I say that? So if you think about the pharmaceutical industry, it really is an information industry. And that's not a unique opinion of mine. The management guru, Peter Drucker, came to that conclusion over 20 years ago. So if we think then that we're dealing with an information industry, why don't we use the latest, where we're dealing with decisions based on data, why don't we use the latest techniques and how then we could create and understand to make those decisions using machines, looking at the wealth of data we now have to play with. What I want to talk about today is to give you a guide, a run through, actually I think of some of the highlights of what Exscientia has been working on. This is how we want to think about using machine learning, AI, actually at every stage of our drug discovery process. How do you understand how you select patients, how you select targets, and actually how you design molecules as well. And that's sort of what I will walk you through today is actually what Exscientia has been up to basically for the last decade. Being a public company, we have to say the forward-looking statements. But what I want to start off with is just this concept that blew me away when I realized this idea. So we're all used to these graphs, how long it takes to discover a drug, et cetera, how much it costs, et cetera. These almost become cliched ways to introduce many talks. But in fact, what's more interesting is how long it takes science to become a new product, a new medicine. There's a really interesting analysis conducted of NIH grants about when scientists first started to publish on a particular area and when that area actually led to new medicines. On average, it's around 30 years. That is a huge delay between you first having your ideas when actually it could impact a medicine. And now we see companies working on Pol Theta, example, in DNA damage repair, still only in phase 1, phase 2. I remember some of that work being published in the mid-90s. So what we need to think about then is how can we accelerate this? How actually do we turn our concept to turn in new ideas into new medical innovations and much, much quicker than we currently do as a society? So the way we think about Exscientia is the concept of sort of shifting the curve. The curve here is this sort of this coastline sort of wave, is to think about the investment and time it takes at the start to need to invest into creating new medicine, et cetera, and then return in sort of profit and revenues from that, and how long before it sort of comes off pattern. As we know, typically inside a pharma company, this can take 10 to 15 years. That doesn't take into account sometimes sometimes 10 to 15 years prior to a project being started before we have enough evidence and confidence that we start to invest into that. The reason that takes so long is that it takes about $70 million sometimes, according to AstraZeneca, just to get to a sort of IND, GLP tox, before you even go into the clinic. So therefore, these are major investment decisions, actually, and that's why academia spends a long time thinking about and validating and falsifying the hypotheses before we actually take something forward. So what we want to do then is how do we, through technology, start to then shift this curve so that we can then start to then bring things forward, accelerate the timelines it takes to go from an idea to IND and from an idea to an approved medicine, and overall then try to reduce the amount of money it takes to actually sort of develop our drug in the first place. If we can do this, we will fundamentally change the economics of the pharmaceutical industry for the better. We model this. So using a whole range of Monte Carlo simulations of various sort of success rates, et cetera, what we find then is that based on the experience we have to date of about 7 drug candidates sort of being developed by our system so far to date that we've announced, the line sort of in orange, we actually see whatever 300% sort of increase in the value, the risk net present value of a particular drug, if we can start to sort of bring down the cost of early stage drug discovery. But an even bigger win is what if we can make an impact into the the time and cost and success rates of clinical stage development. Even if we just make a 5% change to the success rates, we actually create up to a sort of a 600% increase in the RMPV. We believe this is potentially very doable with some of the technology, which I'll talk about in a few minutes' time in this lecture. But this is where we start to see where transformation comes from. It's not just about developing drugs faster. That's really important, of course, because the patient is waiting. There is a real demand for new medicines, but also ultimately better drugs as well. How can we target toward the right patient directly? This fundamentally is what Exscientia is interested in. How do we actually use machine learning and AI approaches to design better molecules, but also how do we identify the right patients for those molecules? And that's the kind of data I want to show you today. So as a company, we are a tech company. Our goal is to actually try to encode and automate what we consider to have been an artisan process of drug discovery. The idea of sort of a drug hunter, the expert, is a very strong sort of image in the company, in the pharmaceutical industry, from Paul Janssen to James Black, et cetera. This idea of sort of a mythical hero discovering a drug. But actually, if we want to turn sort of drug discovery into an abundance, we need to think about how we systematize it. We need to think about how we turn this then into a very reliable process. This is why fundamentally now the technology that sits in your iPhone has been decreasing in its cost year on year, and yet one of the largest drivers of increased costs has been healthcare services in the US, which has increased year upon year on a per. One of them is based on using human experts. One of them is based on sort of processes actually where we can move faster. So what we want to do then is how do we understand what is sort of is a tacit knowledge of how do we make a drug, and how do we convert that then into a process that's reliable and robust. The focus of a company then is to use those processes to design patient-centric drugs. I'll explain what I mean by that now. Drugs targeted for very specific individual sort of patient groups. And we're at the stage now where what I want to show you today is data which I believe starts to validate the platform to show this kind of machine learning-led approach is very viable with some of the first clinical trials we've been running and some of the first sort of molecules we've been designing now in the clinic. So this is what I'm incredibly proud of. Exscientia has now been going for 10 years. Started on works that we published in Nature originally. But we are a company now which I think actually really can stake a claim of being sort of pioneers in the space. We've now got 3 of the first drugs generated by algorithms designed by AI now in the clinic. What I mean by that is the molecules we make and test are generated directly by algorithm. It's not just about producing systems which chemists can use as molecular architects to decide where to put atoms. It's about actually how do you, as you first sort of approach, have the molecules that you make and test be generated by algorithms directly. And that's a key tenet of a company. Also want to show you today actually the first sort of use of a deep learning systems to actually improve clinical outcomes in oncology. How then an algorithm can select which drug a patient should take for a particular individual tumor better than necessarily than a physician could in a prospective clinical trial. And what we've done now is that we've got multiple molecules now heading into a clinic, 3 in IND-enabling studies, 3 entered into a clinic. And what we have now there is data. Data of how these systems have been performing, and I think actually shows a very robust sort of progress, particularly the use of generative design and active learning in showing actually we can create repeatable and I think sort of unprecedented sort of productivity data. So the company has been growing enormously. We've actually doubled in size, I think, every year for the past 4 years now. Today, we're about 450 employees. And what's really important is the makeup of those employees. We call ourselves a pharma tech, and it's the equal importance of technologists and drug hunters. The importance of the drug hunters being able to know and ask the right questions, and then the technologists actually to work with them to help them think of the solutions. And that combination in the culture is actually essential. I think that's what actually, for me, what makes the company what it is. It's its ability to sort of speak both languages, the languages of tech and the languages of drug hunting. We're a very international company. I think we've probably got well over 40 different nationalities within the company. Sites from Oxford, Vienna, Miami, Dundee, opened up here in Boston just recently as well. London, Cambridge, UK, and Japan, Osaka as well. And that, I think, is an incredible diversity of a company. It's actually, I think, is what science is all about. And one of the key things, actually, that leads to being able to recruit talent from all over the world. The company itself, as I said, started from originally sort of academic research. I used to be an academic in Scotland, in Dundee. And unusually, if you know the history of a company, and I could tell you a lecture on this in its own right, how we built a company. It's really interesting that we built probably, I think, one of the UK's now biggest biotechs. And unusually, we avoided VC investment. That's a very unusual way to do it. We focused on deal making and partnership. And the reason we did that is because we thought it was more important to validate the technology on real-world problems. And that was our sort of aim of a company, particularly in the first 5 years. How do you validate your tech in a real world to show it actually delivers? And we did that. And then we started to grow the company, moving from just being a purely sort of in silico-based company, then to building labs. The key thing there was understanding the quality of the data that we needed to drive the algorithms. And this is why probably 40% of a company now are wet biologists in the labs, building out a whole range of different assay technologies. And the key to that is understanding the quality of the data that we're going to sort of build into our models. And then moving more recently into the clinic. And that'll be something, hopefully, we'll be talking about a lot more the next year or so, particularly how we want to use the concepts of sort of adaptive sort of trial design alongside the ideas of adaptive sort of drug design that we used earlier in the stage of a company. I'm incredibly excited by that. So, the key to thinking about successfully using machine learning in drug discovery is to think about what type of problem drug discovery is. In many ways, we're dealing with an unbounded universe to some degree. It's an incredibly complex, multi-dimensional problem that we're trying to solve. We are dealing with a wide variety of different data types. It isn't just about one type of data. It isn't just about high-content data or just about three-dimensional crystal structure data or pharmacology data. It's about all of the above, plus probably another 20 different assay technology types of producing different types of data that we need to integrate into our project. It's also, I think, about data now has reached a point where interpretation is potentially beyond the human limits. And why do I say that? So if we think about all of the new sort of advances that we've had in technology in the pharma industry over the past 2 decades, from sequencing the human genome in the year sort of 2000 till only last week Illumina announced that a sort of a $100 genome is potentially here, to the invention of CRISPR, which is building's almost dedicated to it, to the ability now to engineer cells, our ability to look and understand cells not just in high content, but at single cell analysis and single cell sequencing. The amount of data that we can now generate is astronomical. In the UK, we're currently sequencing 500,000 people's genomes at 10x. That data will be publicly available in about 2 years. Some big pharma companies pay nearly $250 million just for 2 years early access to that data. But the weird thing is that generally, the cost of drug discovery, the cost of bringing a new drug to market has continued to rise, continues to increase. So we're in an industry where we have a paradox of increase in technology, increase in data, and yet our productivity constantly sort of goes down. And I think the solution to this paradox is one thing has not changed, and that is the cognitive ability of the human mind to try to sort of process and understand all this data. If we see now as the pharmaceutical industry as an information industry, we have been overloading the amount of information now that project teams need to think about. If we go back to when sort of Paul Janssen in the 1960s and 70s was running drug discovery projects, and we probably look at the amount of data they were dealing with to drive those projects, it's a fraction probably the amount of data now we ask our teams to look at. So therefore, it's not surprising that actually our ability to generate data has probably been, in fact, we've been running to stand still. The fact we've actually maintained productivity whilst we have a tsunami of data, I think is to a testament of our teams and our skills. But at the same time, that's why I think actually the use of machine learning is so important, because it comes to the crux of what the problem we actually face is, which how do we deal with huge amounts of data simultaneously in a project. And then the other sort of key to it, we believe, fundamentally, is that current disease models have limited translatability from the laboratory into the clinic. 96% of all drugs we will test into the clinic as an industry will fail. We spend something like 83 odd billion sort of dollars a year in the pharma R&D. So 79 million or so of that is potentially wasted if you think of those attrition rates. So this is why fundamentally how we think about disease modeling is going to be key to changing our downstream attrition rates in the clinic. So we then have built a company around sort of certain tenets of how you think about technology and how then you want to build your technology around these. The first one is think of drug design not as a screening problem, but as a learning problem. We are in this paradoxical situation of having a wealth of data that I've just described, and yet on any one project you might also have a dearth of data, small data. You might only have a handful of hits when you start a new project. It's a brand new, first-in-class drug target you're going after. Maybe very little is known about the chemistry of that target. So we have this paradox of both being a small data and a big data problem. So therefore, the key to that is to think about how fast you can learn within a project. The other type, as I mentioned, is learning from all types of data. Don't just build algorithms and models on one type of data, like structure or high content of pharmacology, but think about how you're going to incorporate all those different data types into your models. And a core one for the company, for Exscientia particularly, is how do you generate molecules using machine learning and AI? How actually can algorithms actually do the design and optimization work, rather than actually just providing you with ideas? We actually put them front and center in how we generate the molecules we want to take into synthesis. And actually, I think one of the fundamental ones I want to start off with is the concept of the patient being the best model. If we think about improving the ability to create better disease models, we fundamentally came to a conclusion then we should start with the patient. And the patient themselves are actually the best model of their own disease. So therefore, if we want bioinnovation, we would argue this is where we should start. So actually, rather than start off with describing hit discovery, then describe or target selection, hit discovery, and go to a clinic, I'm actually going to start off with a clinic and how we move backwards from there. So conventionally, we think of sort of drug design and drug discovery as this linear process. We have ideas, we generate new targets, we create new molecules, we move it into the clinic and hopefully select the right patients. But in fact, that linear approach, I think, as sequential problem solving is an inefficient way to do it. We much better thinking of it as a learning process. And in fact, a way to start then is actually with the patient. How do we understand what the patient's experiencing? What's the sort of data we want to collect on the phenotype? How do you collect actual patient sample materials, which we can then conduct a wide range of omics on, whether it's single-cell sequencing or high-content screening, in fact, a multimodal approach, and bringing that data and those models and those assays all the way back then in target discovery, actually conducting target discovery on patient material, then moving those targets then into the sort of the drug generation, data generation sort of processing, and then moving them forward then back into the clinic using personalized medicine approaches, including sort of using the same models again to select our patients and build our biomarker models. This effectively is the architecture of Exscientia and how we sort of been building a company. And what we're doing now is that we're focusing on how we build out sort of adaptive clinical trial approaches, how we build out sort of patient screening tissue platforms for biomarker discovery, how we try to automate as much sort of lab data generation as possible, including synthesis as well, how we move from small molecules even to biologics, and that'll be something we'll be talking about a lot more in the next year or so at Exscientia. And also then how do we sort of select targets based on the patient tissue material coming in. These are sort of the areas where we actually use a wide variety of approaches, data, and machine learning. It's not actually one algorithm. It's actually hundreds of algorithms then positioned at all these different places, points within sort of value chain. But more importantly, think about it as a learning loop. That's the key to this, et cetera, rather than just some ideas moving in a sort of linear fashion. What I want to start off with then is the concept of using the patient to select better drugs themselves. So I want to tell you about a clinical trial we just published in cancer biology called EXALT-1. This is based on computer vision of sort of patient material. And in fact, now it goes well beyond sort of high content. Now we also do sort of single-cell sequencing and transcriptomics of all that data. But the key to it is why do we want to focus on gathering sort of patient material, where it's sort of direct biopsies, malignant perfusions, ascites, where we can try to understand then the range of different cell types, the range of cancer cells, immune cells, and normal cells altogether, is because we want to try to create more sort of relevant sort of samples we can. So one of the problems we have then is that we see much of our research, of course, is based on sort of cell lines, PDX models, and more recently organoids. And of course, organoids, we are investing in, we see a great opportunity for organoids themselves, but even better would be to actually use the patient material itself. Problem with some of this stuff is that it's been really easy to grow, really easy to cultivate in a lab, generate huge amounts of data, but the ability to adopt to the culture and the genetic drift really questions the validation potentially of a lot of this data. Also, it's very homogeneous often. You don't have the range of sort of cells which you have in a tumor. Of course, there are genetic approaches, looking at sort of biomarkers to do this, et cetera. But these are often sort of very reductionist and often very hard actually to go back and try to understand the relationship between phenotype and genotype. And also it's an ability to say, yes, you may take this drug or not that drug, but can you potentially rank which drugs in order you should take? So what we've built then is basically built on a network of hospitals. We have over 70 different sites across Europe where we're collecting sort of biobank samples from sort of various sort of physicians and their patients. Then all the sort of testing to understand between fresh sample and sort of biobank sample, and I'll show you the clinical trial which we've run fresh sample in a bit. See very strong correlations between those. And a lot of our work actually was thinking about how do you sort of do deep imaging on tissue material, which is basically non-adherent cell lines. And this is where a lot of the patterns and know-how sort of reside actually, which is really about how do we then look at those cell tissue cultures, sorry, not cultures, those cell material, which has a wide range of sort of three-dimensional structure to it, often sort of clusters of cancer cells, immune cells, and of normal cells together, and also to understand and then identify the different cell types and the different ways those cell types are being perturbed. This is fundamentally why it's a computer vision problem, and we built a number of proprietary sort of pipelines to actually understand and analyze that data. What we can do then is that we can then do actual individual drug screening on an individual sort of patient tumor sample. So we started off actually by, this is sort of a proof of concept that we did before we sort of run the trial. So we took an 82-year-old patient with diffuse B-cell large lymphoma, And what we found actually then, this patient had failed all other previous lines of therapy. So basically, they were refractory to current therapy and no further sort of treatment options. What we did then is take a sample of that. We then run over 100 different clinically approved drugs, testing each one individually against its response, looking specifically at, does this particular drug kill the cancer cell rather than just kill every cell in the well? And we were able then to sort of rank all the drugs based on their sort of relative sort of blast fractions sort of being destroyed here. Cisplatin came up to be sort of the highest ranked sort of drug of over 100 tested here. But for this patient, given their state, it was deemed far too aggressive a therapy. So actually the next one selected was ibrutinib, which is really interesting because ibrutinib itself had failed a clinical trial against this very disease of diffuse B-cell lymphoma, and this was done actually before our trial was run. So even though that drug had failed that clinical trial, it actually is the potentially the best drug we identified for this particular patient. That patient then was prescribed ibrutinib and a complete remission for 2 years, and actually this is at a fraction of the cost then of CAR T therapy. So what we did then was to run this as a prospective clinical trial, taking this technology, taking individual tumor samples from individual patients, and I believe this is true personalized medicine, and understand then what our system tell you what should be the drug that individual patient should take. So we started off using blood cancers, range of lymphomas and leukemias in this particular trial. And if we look at the sort of control arm, what we find is that within a year, nearly everyone had arm, and it was sort of no progression-free sort of patients within it, et cetera. However, actually, when we looked at the patients for which we'd run the system, and we run the system on both. On the left-hand side here, the physicians decided not to match for treatment. They did not prescribe the treatment recommended by the platform, but in orange here, they did then prescribe the treatment that our platform sort of recommended when doing the sort of this image analysis on the cancers. What we found then is that 54% of the patients had at least a 1.3-fold response. We had an ORR of about sort of 55% in this trial. And in fact, if we looked at the initial sort of drop-off, those are mostly patients with ECOGs 1 and 2. So they're patients already pretty very difficult for them necessarily to take sort of new treatments. Everybody entering the trial had already failed all of our previous treatments. People have been on 4th, 5th, 6th line of therapies before entering the trial. So what we see then is about sort of 25-30% of the patients had progression-free even 4 years out from the start of a trial. So we believe actually this is the first time that a sort of a machine learning algorithm has been shown to improve clinical outcomes in oncology on this sort of scale. And this gives us real confidence then in the concept that if we can do sort of individual analysis on individual patient tumors, we can then actually use machine learning approaches to select the best drug on an individual basis. The reason why we're also interested in this for drug development is it gives us now clinical validation that the methods that we're developing in the laboratory do potentially pan out into the clinic, do potentially lead us to understand that we have higher confidence that actually this then will lead to hopefully better outcomes as we move forward. This is how potentially we shift that curve to higher sort of success rates in the clinic. We now, you'll be hearing about us starting more of these trials in solid tumors as well, because we already started to develop sort of solid tumor models on a range of different cancers using this technology. What we want to do then is to use then the patient, use this sort of patient technology into every stage of our drug development process. How we use it for target discovery validation, how we use it for compound discovery, how we use it for preclinical development to develop new biomarkers, and how we then use it in the actual running of our clinical trials. This is what now we focus on as a company. This is how we want to bring a patient into every stage of the R&D process. So now we've developed models for thinking about translation. We can then use those same models all the way back in the early stage of target selection. So this is some work that was published at the AACR earlier this year. Far more work is going on behind the scenes in this sort of concept. What we want to do here is conduct sort of unbiased screens on understanding how then we could discover new drug targets potentially associated with that disease. So what we've done here is that we've taken the kind of model which we showed earlier for blood cancers, and we've now actually built an equivalent model for ovarian cancer. Each one of those rows is an individual patient, sample from an individual patient. So here we're looking at sort of a target discovery, not just assuming that it's a single disease, but we actually want to look at least 20 different sort of patients and when we think about this. Each of the columns is actually testing an individual drug or an individual sort of chemical tool against that particular patient's sort of experience. What we find then actually, firstly, there isn't actually sort of one target here or one sort of compound or one molecule that sort of kills all ovarian cancer. We often think in target discovery about the concept of sort of this platonic ideal of this drug target to cure this disease, et cetera. When we look at sort of the individual sort of patient response, we look at the diversity and heterogeneity of it. That platonic ideal does not exist. What we're looking for is which drug targets can target particular subsections of that particular sort of patient group who've sort of been labeled with this concept of ovarian cancer. What we find actually then is some really interesting sort of individual responses, as well as some clusters of areas which we'll come on to in a minute. Now, one thing to be careful of are potentially these individual responses here. Look at this particular patient here, has a really strong response to this particular drug here. Imagine that was a patient we took a sample from and then built a PDX model on. We could convince ourselves that actually that drug would have a really strong sort of response against sort of ovarian cancer. But then when we take it to the clinic, find that 95% of the patients then fail to respond against that drug. In fact, I would argue that is the history of cancer drug discovery. In fact, this is why I'm so excited about the use of such a diverse range of patients right at the start, at early stage. We can understand then, here is another drug target. This is what we sort of published a poster on. This actually is ALK. When in fact, about 60% of the patients are responding very strongly. So doing further work now to drill down to find actually there's another pathway behind this, which we haven't yet published, which actually is driving this, which we can infer actually from all of the other sort of data of other kinase inhibitors sitting here. So this then, I think in one slide sort of gives us a philosophy of what we're trying to do. How do we understand sort of patient diversity, patient heterogeneity, right at the start of our drug discovery project? So we can actually then already start thinking about how we can identify these 60% of patients as a population we want to go after. So there are two ways of thinking about using sort of high content and computer vision in sort of drug discovery. One of them is to go for sort of big data approaches, screen huge amounts of molecules and large amounts of sort of cell lines. But to do that, actually, you often require ideas of how do you go for more of a sort of how do you actually then sort of generate large amounts of data by using sort of recombinant cell lines, et cetera? The downside of that, I would argue, is what's the ability and the representation of our true disease. The other way I've just showed you is to actually use a much more focused approach of using patient cell lines. Now, the downside of it, of course, is that we're doing far fewer experiments. We're testing far fewer hypotheses than necessarily a full sort of large-scale sort of compound library screen. So to do that then, we want to prioritize which hypotheses we then want to think about testing, et cetera. And to do that, actually, we use a combination of sort of deep learning and knowledge graph approaches to sort of boil the ocean sort of all of the wealth of literature that's out there. So one way to issues we found, actually, of course, is disambiguating all the different words. The word orca appears in the PubMed many different times. And in fact, using network approaches, you can then work out what actually the different sort of associations of other words around it allows you to then classify that this is talking about a whale, this is talking about a human gene, this is talking about software, and this is talking about a plant gene. So actually network approaches and a knowledge graph are really good ways to help us disambiguate sort of words and understand that. Then once we've built knowledge graphs, we can then start to use sort of deep learning approaches to start to then understand and predict what is the sort of potential citations and publications and linkages between ideas and sort of drug targets. This work I found absolutely fascinating. We published a paper called Trendy Genes, this should be referenced here, about a few months ago. And this is the sort of tip of the iceberg of the work that's been done by this group. So what we're interested in now is how can we use deep learning to generate hypotheses? How do we use deep learning for ideation of new sort of concepts about which target potentially is used to treat which disease? And what we found then is if we look at the range of sort of data and have those interactions between sort of different entities in the knowledge graph connect, we find actually the concept of sort of a new idea, of a new hypothesis, is already present sort of in the latent stage of deep learning graphs that actually even years before it appears in the literature. So the way to think about this is, as a scientist, you would read a paper. You'd read another paper. In your mind, you put together a hypothesis. You'd go apply for a grant. You do the experiment, and then you publish it. And it could actually be several years before you have that first idea, and then your results potentially being published as you falsify that idea. So what we find then is that actually, if we took, for example, the whole sort of human genome, we take sort of about 20,000 genes, we then look at data even from 20 years ago. So I'm only looking at data to do with sort of non-alcoholic fatty acid liver disease from 1999, pre-2000. So I take all that was known about NASH up to that point, and then I rank all 20,000 human genes and say which ones are not yet published, but have the highest confidence that they could have some relation to NASH some way, linked to pathways, linked to sort of other ideas within it. What we found then, if we look at the top 100 genes, over the next 20 years, 40% of those were then published in the literature, making a link between that disease and NASH. I find that incredible. What it's suggesting then is that the concepts of sort of what was in the latent space, actually already there's enough confidence around those ideas to make that suggestion that this target should be linked to this disease. And actually then we can potentially predict that years before it's published with some, quite a high degree of confidence. So we're now looking at this for obviously a wide range of diseases and using this in our work with people like Sanofi and how we want to target and identify, novel sort of drug targets moving forward. And for me, I just find this incredible that actually this would be a very interesting way of thinking about how you'd apply for and rank grants, potentially about how novel an idea is, whether that idea is potentially predictable and we should do work to falsify it, or whether that itself is a novel idea itself, which can't be predicted by AI. So we've shown how we could take a patient and we've shown how we could then create sort of disease models. We've shown how we could use those disease models then to generate new target ideas. Once you have a target idea, how then can we use machine learning to create a new molecule? This is what I want to talk about next. The concept of sort of generative design. The concept that actually medicinal chemistry can move forward from just being sort of a work of molecular architects, where we analyze the data, we think about what to make next, we're effectively sketching things on whiteboards often, or working in sort of three-dimensional sort of environments, to the point where actually it becomes a process where actually chemical designs are generated and prioritized by the algorithms themselves. So the way we think about this then is we want to understand and define the project objectives. We want to develop a design system that can use a wide range of different data types, whether it's three-dimensional structures or whether it's pharmacology data or both, and think about actually what might be sort of even a small number of starting points to kick the process off. The process then is about sort of building models, and that is a huge part of the process. Generative design technologies, which have been refined over sort of the past 10 years using a lot of medicinal chemistry knowledge. Active learning, I think, is the sort of young-sung hero of this whole process. And in fact, when I show the productivity data for Exscientia, active learning, I think, has probably been the absolute key to sort of driving that. And more recently, chemical synthesis and biological testing, which, of course, we're actively interested in how to automate it. And I know here at Broad and MIT as well, you're actively interested in that as well. So what we want to do then is firstly think about creating what I would call a data agnostic or model agnostic approach. If we just think about using three-dimensional crystal structures, and then we look at our target product profile, you will find that there are dozens and dozens of other properties that we need to make a drug beyond potency, which are not yet modelable necessarily using physics-based systems. So what we want to do then is to think about how we can then use all of the different sort of assay data that we collect and generate a wide range of different models. Because for many of these different sort of data sources, we will find actually different types of models actually can build sort of a better system. And then actually using sort of this whole sort of cloud of different models, including physics-based models, which you can then integrate into this as if it's another model, to then use generative design, both 2D and 3D, select then, using active learning, which molecules to make and test, because ultimately, making and testing a molecule is still the rate-limiting step. It still can cost, tens of thousands of dollars to make a compound and get it tested in terms of a wide variety of assays we want to test it against. So therefore, every decision we make is an important decision to ask. So the key to this then is building sort of an ecosystem of models, and there are a huge number of lectures, and we could have a whole day symposium about how then we would select data, how we'd integrate data, what are the best practices for doing so, et cetera. What are the best ways of not just building models, but building sort of a whole ecosystem in terms of the latency time, the best engineering approaches to doing this, et cetera. What we want to aim for, actually, is to generate thousands of models from all of the different sort of valid endpoints that we can generate data from, but also generate a wide variety of different models. Don't just build a Bayesian model, don't just build a recursion model, don't just build a neural net model, but actually try to build as many different models as possible and then benchmark whatever is the particular best model for that particular problem at that particular time. And that's the way we move in towards in sort of the model ecosystem that we build inside Exscientia. The key to it then is to link sort of your design to your objectives. This is actually one of the key drivers of what we think we've had some success in generative design. It is actually sort of mathematically defining what your sort of target product profile is. Defining that then mathematically is a set of multi-parameter optimization techniques. We call this a reward bundle. This then is a piece of maths which any of the algorithms they can call against. So everything has been scored against the same set of sort of multi-dimensional sort of target product profile properties and models that we want to build. And then actually to understand how your project is progressing. This is what a sort of typical project looks like. So what we do in measuring here is sort of the x-axis is the number of compounds we're making sequentially. The y-axis is a sort of a multi-parameter metric sort of score reduced down to sort of one number, call it a merit chart. And what we see then is that when you start the project, is that you might be scoring very low. Doesn't mean you're not hitting important molecules. What we're trying to do here is design against multiple dimensions. So you could actually score low because you're failing on one dimension. And then actually as we start to learn through the exploration phase, where we could be testing many different types of chemotypes, move to the exploitation phase where we now sort of hone down to just one or two chemotypes. And nearly every compound we're making now is scoring very highly then on the MPO chart. And in many ways, if we don't see this kind of trajectory inside our projects, we know there's a problem. So the key to what we've done, from evolutionary techniques that we published in Nature 10 years ago to much more recent techniques using of reinforcement learning, is actually the concept then of how do we then sort of use generative methods to allow us to explore chemical space much more widely. A big part of this actually is how do you incorporate medicinal chemistry knowledge into these algorithms so that the molecules that you're generating look, feel, and smell like the sort of molecules medicinal chemists would actually want to make and test. This has been a key thing to sort of cultural adoption as well. If you're generating molecules which come out as sort of being unsynthesizable, the chemists will stop using your algorithm very quickly. So this is one of the key things actually is sort of understanding and testing these with sort of expert chemists as you go along. In fact, this is a key role of chemists inside Exscientia, is how do you bring that sort of med-chem tacit knowledge into making sure that the generative algorithms actually are generating chemically sensible sort of starting points and designs. So what we're ultimately trying to do then is to think of how we build a whole range of different models on all the different properties that we want to model, define those as a set of objectives from our target product profile into objectives then that we learn against. And this is an example then of a reinforcement learning algorithm. In green are sort of the parts of a molecule which are then scoring highly. In red are the individual atoms which are scoring lowly against the MPO. And over time, we direct then the evolution and generation of that towards then generating most of the molecules then that score highly against the MPO that we're looking to optimize against. This is a sort of general sort of flow chart. You can actually start off by using both 3D and 2D generative methods, if you have a crystal structure or just using pharmacological data. This might be, for example, a reinforcement method. This might be a sort of evolutionary design method. And what we're trying to do then is to then filter and understand different model space. So we still use the concept of evolution really strongly in our design. The concept being you have a generative process where we want to generate lots of ideas, and then the models providing a selection pressure to actually then select out the best from that. And then end up with an elite population then for further refinement. And the further refinement we often use and found to be incredibly successful has been active learning. The idea then actually, we want to understand as we starting off from a sort of understanding sort of the binding site using things like hotspot models, generating models in 2D and 3D, thinking about synthetic tractability, And then the key to it then is potentially late stage using some dynamics as well, but also active learning. So active learning is a concept of which experiment should I do next mathematically? Which experiment actually will give me the most information to build a better model? And this means potentially making compounds which are potentially inactive even, or not necessarily as potent that you would normally select. So one example of this I hope to talk about publicly in the next few weeks and months. One of our projects has been a concept actually of what we're looking for, and is not necessarily selecting models on the Pareto front, but active learning, actively allowing us to break dogma, suggesting ideas for compounds in chemistry, which allows us then to sort of understand new chemical synthesize and come up with new rules which weren't possible within it, actually. So the other thing as well within that, actually, once you've defined your project, once you've run it through sort of several design rounds, is also then to use mathematical approaches then to understand how your project is progressing. What we found here actually is concepts of understanding sort of our predicted sort of MPO scores against understanding the certainty we have in predictions or the uncertainty we have in our models. And what you find actually then is as the project progresses, not only should we be making better compounds, but our models obviously mathematically should be getting better as well. And this way we can measure how much we're learning within the active model space. What we find then actually can allow us to understand where we sit in sort of on this grid, understand whether or not the project is we're learning more, but actually we're not progressing, or actually we're sort of progressing, but we're not necessarily, our certainty is still low, to get to a point where we can now nominate a drug candidate, where we have high scoring molecules and we have high certainty in sort of the scores that we can predict. This then is a really interesting way then of thinking about, you don't just keep going on a project until you think you're the best molecule, or ask like, is there something better again? But actually try to mathematically define when good is or when good enough is. And that actually can also save a huge amount of sort of time and effort in knowing when to stop. Very short of time, but I'll quickly run through sort of last sort of case study, which is one of our molecules in the clinic. This is with some of the older tech that we were using, but it's still sort of a good example of an A2A molecule for immuno-oncology. The key we wanted to do here is design it so it wouldn't get into your brain, and design it to be highly selective for A2A, because we'd seen data that suggested to us that targeting A2B also led to sort of a decline, sort of bell-shaped pharmacology, which we wanted to evolve. The molecule itself was designed using generative methods from fragment screening against wild-type GPCRs using SPR. It took only 9 months to run that design project, and it was only the 163rd novel compound that we made and tested. Defined was sort of merit charts early on about what sort of ideal sort of profiles would look like for the MPOs. And this is the MPO then of a merit chart for our project. For 2 different colors, actually, sort of 2 different sort of chemical series. You can see a second series was found and then actually allowed us to really sort of create a lot of high scoring molecules. What was interesting about this, we use a lot of biophysics at Exscientia. Nearly every compound that we make and test where we can, we run sort of kinetic profiling on using SPR. And I think we were leaders of using this against wild-type GPCRs. So basically, we're not thermal stabilized receptors. These are the native GPCRs. And therefore, we can run this across all of the different GPCRs, all of the adenosine receptors, and all of the different sort of species type. And what we found then was some new chemotypes, rediscovered all the existing chemotypes using this, and allowed that then as the seed for sort of the generative algorithms to move forward. There's an example here of how that sort of process is working. Using not only sort of existing PDB structures at the time, actually. What we're trying to do here is not just optimize against one property, such as potency, but trying to optimize against all the properties in different merit score with models built for each of the different sort of top properties like solubility or selectivity properties, etc., or CNS properties that we wish to sort of optimize against. What we're trying to do then is to optimize the total set of properties there within the merit score. Push that down, pushed a lead molecule in cycle 4 then, led to sort of a breakthrough on the project. Pushed that down even further and allowed us then to identify through generative design one of our little gambit, particularly this regio-selective sort of concept here. And that allowed us to really drive through the project into a new sort of subset of this particular chemotype, which then all the molecules in green are sort of a new subset, which then led to the drug candidate. What was interesting then was how the properties of those molecules sort of compared then against the competitor molecules, much better sort of avoidance of the brain, and much better sort of selectivity, we believe, over A2A. And the phase 1 in humans suggested that the predicted PK matched what we have expected. And what's really interesting, we'll be announcing very shortly how we're going to run the phase 1B in patients. And here we've gone back to the tissue patient platform I mentioned earlier. And what we're seeing now is that we really see that the use then of this platform also potentially for immuno-oncology, because we also have the immune cells next to the cancer cells within the sample, allows us then to understand how we can sort of perturb and understand which patients potentially could benefit from this technology. And we'll be making more announcements on that in the next few weeks. So, using generative design and active learning, we have run this now multiple times. 7 drug candidates, 4 in IND, 3 entered into the clinic, more to be announced in the next few weeks and months. What we see now is a real sort of robust process. On average, we're making around 250 molecules from an idea to a drug candidate, and compare that to the industry average, which is about 2,500. We did an interview with Frank Nestle from Sanofi, head of R&D there, earlier this year, and he said Sanofi would often make 5,000 compounds in a project. And what we're seeing then is that some of our projects is even lower, sort of 130 or so. So this is where we believe actually the concept of generative design and active learning is really starting to show real results in the real world. And of course, if you're making fewer compounds, the projects generally also tend to run much faster as well, because ultimately we're doing fewer experiments. And this is where the concept of it. Obviously, if you're working on new targets, do expect to see this early stage increase a lot more as you start to understand and validate your biology. And of course, depends on the post-IND studies as well. But in terms of sort of drug optimization, we believe this technology now is starting to show us real data about how machine learning in drug design actually can make a difference. And just to finish off now, just one last story. One of those molecules who are heading into a clinic is CDK7. This is a non-covalent sort of inhibitor, exquisitely designed actually with its PK so we can institute drug holidays. The competitive agents were not very selective, particular issues with gut reflex. We designed then a novel compound in less than 12 months, less than 136 sort of experimental compounds. We believe to be potentially a best-in-class molecule for this target. Not only we got the traditional PDX models, which look great, but more importantly then, how do we use our sort of design engine? How do we use our translation models to help select patients? What we can do here then is to take 10 individual patients, and we can then run against the ovarian model we showed earlier. And what we can understand then is about 60% of those patients respond in a very nice dose response, and the other 40% for second cohort actually respond at a much higher sort of dose compared to this. What this allows us to do then, pre-clinically, before we go into a clinic now, is to identify which cohort we believe we should be selecting for our clinical trials, and also then to go back into sort of the multimodal sort of phenomics and genomics approaches to now identify what are the sort of genomic biomarkers that distinguish between these two populations, and then build those into our clinical trial. We're incredibly excited by that, because this then is how we want to design our clinical trials going forward. And with that, I wanted to thank you all today, and I wanted to end by saying that fundamentally, we believe all drugs should be this way. I'm incredibly proud of what we built at Exscientia the past few years, but I also believe that we now have a huge community of people represented by this audience who are interested in this concept. How do we challenge the way drugs are being created? How can we use information and data to create a new way of creating new medicines? And I believe fundamentally is I think everyone in this room envisions a world where we want to create medicines to be in abundance, not just to be a scarce resource, but actually how are we going to tackle those potentially tens of thousands of orphaned and rare and importantly now sequential diseases. Now we think about cancer not as one disease, but as many, many diseases. The only way I can do it actually is the application of this kind of technology, and how, as a community like this, we develop it further to create a world where drugs become in abundance. Thank you so much for your time, and I really appreciate it. Have a good day.
Thank you, Andrew, for that fantastic talk. Very inspiring. We're really impressed by what you've been able to accomplish. Do we have any questions? If so, could you please come to the microphones?
Hi, Andrew. Nice to see you again.
Good to see you again. Hi.
John Davies, Totus Medicines in San Francisco. So I love the fact that you laid out a beautiful overview of early research work in terms of machine learning and AI. What can you say, though, if you with the regulatory bodies of Europe or the FDA in the US about what needs to happen on their side to increase their receptiveness of machine learning and AI in clinical?
That's a great question. So at the moment, we are all, as a community, working within the existing confines. When we take a drug forward, whether or not it's being generated by algorithms or whether it's designed by humans, we still work through the same regulatory framework. When we take a trial forward, we still work through the same regulatory framework of how we want to design that. So what we're not doing here is necessarily challenging the existing frameworks. You know, I think we're still in early days and we want to play by the rules and be members of a club, so to say. But I think there are some really interesting ways how we could start to think about how we would, you know, where actually regulatory innovation might really be useful. I think the Exalt-1 trial was a very interesting sort of way of thinking about, if you look at that data, I ask myself, how could every cancer patient benefit from that? So right now we are using that, you know, in the drug discovery process, as I showed on the last slide, how we would use that to fundamentally select which patient cohorts a particular drug should go into. But we believe it has got potential well beyond that. And that's interesting. So the questions we ask ourselves in-house is that a diagnostic or is that a digital therapy? When the algorithm is actually selecting the drug to move forward. And I think that's a very interesting question. We also have questions then around how would you sort of potentially monetize, you know, that kind of technology to move forward. So I can't give you an answer yet, but I do think actually we are in a position where we could think about where is the boundaries then, particularly I think between therapeutics and diagnostics going to come. And I think that's probably the area probably richest seem to mine for our interactions with regulators. The other one actually, which we'll talk about much more sort of towards the end of the year going forward, is the use sort of adaptive trials. And potentially then when we want to think about sort of ideas that go beyond the traditional phase 1, phase 2, phase 3, to a trial becoming continuous, where we're designing trials not just to learn about the drug, but also to learn about the types of biomarkers we're starting to describe. Because actually validating our biomarker is going to be equally important as we think about what might be the companion diagnostic for that drug as well.
Samaya, do we have any questions from online attendees?
Yes, plenty.
Good to
So I'll go ahead and ask the first one. Do you see FDA-approved compound repurposing drug screen for new biology target and improve the compound designs? Not sure if that's a question, but yeah. And then the next part of the question is, now with iPS technology, we can directly make disease-relevant human cells and test these compounds. Can we do that? Thank you. And
yes, so we ourselves are starting to think about how we start to create organoids directly from individual patients. So you'd think about those 20 sort of patients, we were doing a sort of variant studies, how we'd create sort of 20 organoid lines potentially from those. So then again, I want people to move away from this idea of thinking about cancer as that one cell line you're selected from one patient, and now thinking about the diversity of sort of patient experience and testing multiple different sort of cell lines simultaneously. Coming back to sort of repurposed drugs versus new design drugs, I think that's, if a drug is valuable to a patient, it probably doesn't matter where it comes from in many ways. And in fact, the Exalt-1 trial, you could argue is large-scale drug repurposing. You know, we are finding new uses and uses which are not approved potentially for that patient, and that was the drug to test it. So that's an interesting way to think about it. We ourselves, as a company, focus on the creation of new medicines. I believe actually the use of machine learning really is to design precision engineered drugs. And I think Pat's work, hopefully talk about later today at Relay, is a really good example of that kind of approach. And I think that's where the excitement for me lies as well, which is how do we create precision engineer molecules? I used to actually set up sort of drug repurposing at Pfizer many years ago. We called it indications discovery then. How do you find new indications, discover new indications for drugs? And what we always found was we could make that link between sort of that target and that disease, but the molecules were never had quite right properties for that new indication. And that's why I believe we need to precision engineer medicines for the right use. Sorry.
No.
Thank you.
Good.
Hi. Stephen Jerome. Excellent talk. Really enjoyed it. I had a question about the last topic of the presentation, the use of generative design.
Yes.
So, fundamentally, this is, of course, fantastic technology. But one of the things that we've certainly struggled with at Schrodinger is that, building robust models for some of these data endpoints for which, getting data to train the model is very expensive, right? And of course, then if you're data limited when training models for these endpoints, the domain of those models is going to be limited, right? But of course, when you're applying generative design, you want to explore, you don't really want to be constrained. So I guess the first part of my question is, with regard to training those models, this number of compounds synthesized in the program is very impressive. Does that include compounds needed to develop and validate models, local models for those properties, or is there additional sort of pre-development? And if not, are you mostly using global models that are later retrained or refined during the active learning process? I mean, how is that working?
No, that's a great question. So that includes all the compounds up to the drug candidate being identified. So we're not making any extra models or libraries to build a model, so to say. What we do find is that sort of embrace the fact that your model could have very low prediction at the start of a project. Even if you start off with a global model, because you're in a bit of novel chemistry, by definition, you could have low productivity. And then actually to use active learning to learn your way in, and then about 100 compounds in, we're starting to find then the models are much higher predictivity than at the start of a project. So it's that sort of learning around that local model as you go forward within it. That's key. I think the other key sort of philosophical approach we took was to split generative design from models. So generative design is its own model. It has its own model around, and it's really focused on sort of chemistry and understanding how to explore that. So firstly, it's understanding creates high-quality chemistry, high-quality molecules, a lot of sort of input from medicinal chemists around it. And in fact, when a chemist still have ideas which are not being generated, the first thing we ask is like, why isn't the generative model producing that, for example? What's missing? So that was sort of key to that. In fact, we can sort of regenerate all of Kemble now from just a few key seed points, and we know then we can explore chemical space, very effectively. But then the other key to it, as you said, is that what you really want to do is explore new areas. The really exciting stuff is where your model's scoring low, because it's outside of domain applicability, by definition is novel. And then that's where active learning has been key. So I think you really need to think about sort of generative design, active learning in its exploration space, then model building, almost in that order as the key to philosophical approach or thinking about it. Because particularly when thinking about sort of new chemotypes, and particularly when breaking dogma on a project, there'd often be a dogma that says, all compounds need to look like this. And if you're building towards high-quality models, they will pull you back to that space. But you deliberately need to jump outside that space, and that's, again, why we found generative techniques like evolutionary approaches and some of reinforcement models allow us then to make that jump outside of space. So it's that jump outside of known space which has been the more sort of fruitful in terms of use of generative models.
Okay, thank you very much.
We have plenty other questions from online, but we'll skip it just to keep in time,
Yes.
and we'll take one last question.
One last question.
Hi, Tudor.
Yeah.
Really, really great talk. Much appreciated. On your conceptualization of oncology targets, if you look at literature, 2 out of 3 proteins are associated with cancer, let's say 14,000 proteins, but only 300 are in the hallmark of cancer. Okay. If you look at your merit versus uncertainty score, I really love that plot. That's for compounds. Do you have something similar for targets? Like in oncology, do you have like a merit versus certainty for oncology to try to precision medicine that way as well?
That's a good question. And I skipped over just in 1 or 2 slides just the work that's going on in using deep learning for target prioritization. And yes, confidence values are actually vital to that, because otherwise you would end up with far too many hypotheses to show. We haven't yet sort of fully published all that work, Tudor, and now I'm a public company. I can't fully disclose it all yet. But yes, that idea of understanding the confidence limit rather than just everything. And I think that's fundamental to us because, as you said, half a genome might be associated with cancer more broadly, but it's specifically that is. But yes, we do think of confidence as the key driver to prioritization.
So how do you rank that? How do you score those? Or you can't talk about
So I can't talk too much. What I can tell you is it's to do part of the linkages within the latent space of a knowledge graph is what fundamentally is driving that confidence score as well. So you sort of get the confidence as well as the hypothesis out of the same method. Thank you.
I think we got lucky here, and we can take a couple of more questions from online. So the next one is, in silico structure finding to real chemical synthesis is a difficult process. How you know that the predicted chemical can be synthesized or not?
I think that's a great question, and Connor here as well could give a great talk on that. So it's an active area of research for us. I mean, we've used statistical techniques previously to understand, is this likely to be in synthesizable space? But I think where the field is moving to now is much more definition of actual retrosynthetic pathways per molecule. We hope to talk more about that. We're actively involved in this. We're making a very large investment into building an automated chemical synthesis lab in Oxford, building out 26,000 square foot just for automated chemistry, which we see potentially as the future. We think the future is designed by AI, made by robot.
Thank you so much, Andrew. So, and this is a question from me. So what as a ML community or ML in drug discovery community we are now not doing that we should do in future?
Oh, God, it's a great question. The first thing is just how many problems there are to solve. This isn't about creating sort of one algorithm to rule them all. This is about actually the concept of that sort of graph I put up of sort of the learning loop. Is that when every sort of part of that, from sort of target selection to understanding the value of the data that's within our cells and patient materials, to understand how we design drugs, to understanding how then we would sort of manufacture and those sort of molecules, is there is actually a huge number of problems to continue to solve and refine. I think one way to think about ourselves as a community is ultimately we want to sort of, our enemy is the status quo. Our enemy is actually the normal way of doing drug discovery and development, which has led to, drugs being a sparse. We make 10 times as many movies as a society than we do drugs per year coming forward. And they probably cost as much. Movies these days cost hundreds of millions to make. And yet, so I would say actually one of the key to it actually is to understand to each other as well all the different opportunities within that space. So we don't just all hone in towards one particular problem, but we understand the diverse range of problems that we can then sort of spread our efforts across. Sorry.
All right, thank you all for the questions. Let's thank Andrew again for his fabulous talk.